so much, Darren, and the Andes. I thought that was quite a nice job with the keynote as well. So we're thrilled to, on behalf of the firm, Wilson Sonsini, I'm Matt Bresnahan, a partner in our life science practice, and we're thrilled to again support the First Look showcase as well as the Alliance, which we have for many years now. So I'll be moderating the track, and what we'll have format-wise is very simple, 3 minutes for the pitch presentations from each group. And then 9 minutes of Q&A with our judges. So with that, I will turn it over to our 3 judges, Seth, Pascal, Ryan, to introduce themselves. So why don't we start with Seth first?
Thanks, Matt, and thanks everyone for joining us here. Very excited to be part of the presentations today. I'm Seth Lieblich. I'm a principal at 8BC. We're a multi-strategy fund, and in the fund, I focus on our bio-IT investments that cover a range of areas from therapeutics to platforms to diagnostics and pharma infrastructure.
Great, thanks, Seth. Pascal.
Hi, everybody. I'm Pascal Crotty, and I'm a senior associate at Rivervest. We have offices in both St. Louis and San Diego, and we do med tech and biopharma investing.
Fantastic. And Ryan.
Hey everyone, I'm Ryan Witt. I am the Hub Director of UCLA's MedTech and Digital Health Innovation Hub, but more relevant to this, I'm a member of Tech Coast Angels and a periodic angel investor. Tech Coast Angels is one of the largest angel investment groups in the world, actually. We've invested over $19 million in 64 companies just last year, and primarily seed stage. So very active.
Excellent. Thank you, Ryan and Seth and Pascal. We're thrilled to have you all judge and provide feedback to the companies. So with that, I think we can move on to our first, and we have a lot to go through today. So we'll keep it moving. So the first group is Ericari Biosciences from Uc Irvine.
Okay, good morning. Thank you for the opportunity. My name is Mingyi Huang, CEO of Aracari Biosciences. I'm really very excited to be here. What is Aracari? It's about prediction. We are a better predictor of drug efficacy and drug development safety for drug development purposes. And how do we do that? Because with a human relevant breakthrough technology, we can actually grow in our system human blood vessels, human tissues. More importantly, we can deliver nutrients, therapies, specific human immune therapies, and also take out the waste in our system. And what's the market needs for this? Today, as you know, over 90% of clinical trials fail. That's why it costs so much money, billions of dollars, a lot of time, lots of years, over 10 years to deliver a treatment to a patient in need. And why is that? Because the current animal models, the current 2D models, spheroids, are simply not working that well. And our models are human relevant. And let me just share with you our product, what it looks like. Just give me a second. So right in the middle of this is what we call our product VMO-VMT, vascularized microorgan or vascularized microtissue. And just as a side point, our products have been validated for its reproducibility by an NIH-funded testing facility. So in here, what is this? It's in our diamond chamber. You have a vasculature network, wrap around tumor, cancer tumor specifically in this case. Just like a human blood system, you have the arterial that feed the nutrients, and then you have the venule coming out, taking the waste out. And you can probably imagine you can take all the biologic markers that you need and observe it at different time stamps over time. And the other thing probably you also know is most human cells are less than the width of a hair from a blood vessel. So the application is tremendous. So just to recap, our VMO-VMT system is a 3D in vitro microfluidic device in which living human tissue can cooperate with perfused self-assembled networks of human blood vessels that can supply nutrients, therapies, and remove waste. So market size, as I mentioned earlier, it takes so much and doesn't work in current model. So today's market, animal market alone, animal testing is $18 billion a year just in the preclinical phase. In terms of in vitro tox testing, that's another $10 billion. So as you can see, the market potential is tremendous. So that's our product. So a little bit about Ericari Biosciences. We are a California C Corp based right in Irvine. It's as a speaker mentioned earlier, it's an innovation hub for sure. And we have a seed funding. We also have exclusive license to the technology developed by our scientific founders who are all UC professors developed over the last 20 years or so. More importantly, the last 2 years, since our founding, we have developed loyal customers, we're developing returning customers, and we have positive cash flow, and we are simply looking to really accelerate our growth to produce what our customers have been asking us for is organ-specific models such as liver, blood-brain barrier, and bone marrow models. And the next thing is really accelerate our growth into doing a validation model for precision medicine. So with that, I also want to just give you a brief overview of our team. Our team is actually very talented, very young, 3 major scientists in our lab. They all have years of experience, PhDs. We also have the 3 scientific founders from the UC system. And then the management team is also very experienced. Our chairman, Wes Hetfield, is a very experienced biotech entrepreneur, and myself is very experienced in finance and merger and acquisition. So with that, I'm going to just, if you're interested, please talk to us. You can contact us at this email address or just contact us through the website.
Great, thank you. Very nice presentation. So we'll turn it over to the judges. Would anybody like to go first?
Yeah, before we do that, I just want to maybe introduce my scientific officer who should be online.
Sure.
Chris Hughes.
Hi, Chris Hughes. Yeah, I'm a professor at UCI and the Chief Scientific Officer of Aracari Biosciences. Nice to meet you all.
Yes.
Great. Yeah. Thank you, Chris.
I'll go first
okay.
as a UCI alum. I have to go first and say thank you for this work. It's wonderful. I 2 questions, I guess, to kick us off. One is how does someone from AIG and finance get into a business like this and get attracted to do this? I think often when we're investing early stage, we're betting on the team. Sometimes you have people who don't come from science, which is fine, but looking to understand what's behind that. I think also looking to understand what I love to understand more about who's paying, who are some of the paying customers, and you talked about recurring customers. That's always a positive thing that every angel investor can digest. Would love to hear more about that. Is that Pfizer or those research institutions?
Sure, I can answer those 2 questions. First, that's a very good question. How did I jump from the Wall Street to the innovation hub in Irvine, California? I guess it's part of my passion is really about scientific development. How do we really cure diseases? And just really through networks of people, I got to know the scientific founders in UC Irvine, and they're really looking for someone with business background to help run the company. So that's where I come in. And I think we have a proven track record in the last 2 years now, as you can see, returning customers and positive cash flow. That's what we're being successful at doing. So we're getting ready to the next phase to raise additional fundings, as I mentioned. The second question is, what you call that? Can you remind me what those questions again?
Our customer base.
Oh, yes. I can just mention some of them. I think we say top 10 pharma companies. If you name them, they're probably in there. I can't name their names yet, but because they don't want us to share the names. But if we would talk and have further discussion, we're happy to get a referral from them directly. But they're top 10 major pharma companies. If you go to Google, they're there.
Yeah. I think 6 of the top 10 we're working with right now.
Depends on which list. Yes, exactly, Chris.
Thanks
Does that
so
answer
much.
your questions, Ryan?
Thanks so much, Ming, for the presentation. I too am a UCI alum, so it's always good to see everyone representing. So, I'm glad, you guys are working on another microphysiological system. I know there's a lot of shortcomings on those, but could you highlight for us, what the key advantages are of Aracari over, some other names that have been around for a while, such as Emulate and Mimetus.
Yes, I'm gonna
Yeah,
let Chris take that.
I'll take that one. So all do offer some version of vasculature. Emulate has a very simple version, which is a single tube representing a vessel. They're probably the best model is their lung model. And then Mimetus has something similar to us. It uses slightly different technology. What I can tell you is one of the reasons we are getting so much business is that our customers have tried both of those companies. And I don't want to say too much that's rude about our competition, but let's just say they're much happier working with us. The advantage,
I think, actually, as I said earlier, our products have been validated for its reproducibility by a third party.
Right, so I was just getting to that. So the key here is reproducibility and can you get a project done on time? To get it done on time, it has to work like you say it works. And that's one of the key things we've got. We've put a huge amount of effort into making this reproducible to the point where when we gave one of our customers their data on time, they were shocked. They were like, "You've done it? It worked?" I'm like, "Well, yes, we said it would take 3 months and it took 3 months because our platform does what we say it does." And as Mingy mentioned, we've been independently validated by a tissue chips testing center, something that NIH set up. So a couple of the scientists from Texas A&M came, we showed them how it all worked. They went back to their lab and they reproduced everything that we published in one of our seminal papers on this, on drug responses in tumors. So they were able to reproduce it all and that's been published. So it works, it's reproducible. So that's a key selling point for us, for our customers. But the other one that everyone offers, is that it's physiologic. And that is a key element here that is unique for us, I think, is that our ability to reproduce a real tissue. Everything that gets that tissue gets there through blood vessels. And that's how your body works. And if you really want to understand how a tissue works, how a tumor works, how an organ works, you can't just pour medium on it and expect that to represent how an organ works. Organs work because they take stuff out of the blood, they put stuff into the blood. So we capture that. And so what we find is that when we test drugs, we get data that reproduces what you see in a mouse or a person, but you cannot get that data just with the standard 2D culture systems that most people are using. So we're highly predictive for what happens in vivo. That's the major advantage of our platform.
Great. Thanks for that. And thank you, Ming, for the presentation. Very great. And unfortunately, I'm the only one on the panel who's not a UCI alum, so perhaps I get negative demerits on that. Thank you. I had a 1.5-part question here. So when we think about, or at least when I think about organoid systems, I view sort of two of the major challenges here as being one in 3D organization of multiple cell types in the right orientations, and then secondarily, and specifically when it comes to the endothelial cell types, is generating the organ native endothelial cell types for that particular tissue that you're trying to replicate. And so maybe you could speak to those two areas and how you're addressing those.
Yeah, absolutely. So you're right. The three-dimensional organization of the tissue is often critical to its functioning. What we do, so let me say, some people, what they try and do is they try and engineer their way to a solution. And that's fine. You can sort of pre-pattern everything. What we do is a simpler and I think more physiologic version, which is we put everything together and let the cells sort it out. So the biology is incredibly powerful. Cells have an amazing ability to self-assemble into tissues if you give them the right conditions. That's what happens for us. So for example, the blood-brain barrier model, we get tubes of endothelial cells. We get cells called pericytes wrap around those. We get cells called astrocytes that kind of put down these foot processes onto the vessel. That helps to develop the vasculature in the brain. We see all of that happening. So you're correct, that three-dimensional structure is really important. We see that in the other tissues that we create as well. Your second question about the vasculature is a very insightful one. We're just about to publish a review on that, actually. So the vessels, blood vessels in each tissue are kind of specific to that tissue. So heart vessels are different to brain, different to liver. How that develops in vivo is crosstalk between the tissue and the vasculature. The blood vessels help to pattern the tissue helps to pattern the blood vessels. So again, what you're talking about is a cooperation between cells. And again, that's what we see. We put in cells that are what's called plastic. They have the ability to take on different types. So the cells that we use, they're not pre-committed to being a particular organ. So when we put them in our brain system, they take on the brain type. When we put them in the liver, they take on the liver type. When we put them in a tumor, they take on a tumor type. So again, what we do is put everything together and let the cells create the environment that they want to be in. And we find that's a very powerful way of doing it.
Yeah, excellent. And thank you, Chris. And judges, thank you for the questions there. Me and Chris, thank you so much for the presentation, and best of luck to you.
Thank you very much. Thanks for the time.
Thank you. Next up, we have Myogene Bio from UCLA.
Thanks so
Courtney,
much.
Yeah, go ahead.
Can you hear me now? All right, perfect.
Yep, we can.
I'm Courtney Young. I'm one of the co-founders and the CEO of Myogene Bio, which is developing cutting-edge therapies for muscle diseases. Our first approach is for Duchenne muscular dystrophy, or DMD, which is a really horrible muscle wasting disease. It leads to wheelchair use in the early teens and typically premature death in the 20s or 30s. And there's currently no cure. This photo on the right is my cousin Christopher. He has Duchenne. He'll be starting high school next fall and is an avid chess player, but he's going to die in 10 to 15 years without a therapy. And so he represents the patients that inspire us to develop meaningful treatments. In Duchenne, there are genetic mutations that result in a lack of the dystrophin protein, which is what leads to premature death. We've developed a solution called Mimetus 4555 that uses state-of-the-art AAV delivery of CRISPR-Cas9 to use gene editing and permanently remove a hotspot area of mutations for around half of all Duchenne patients. This will allow for dystrophin to be produced, which is expected to significantly improve the lifespan and quality of life for Duchenne patients. Duchenne affects around 1 in 3,500 to 5,000 boys annually worldwide. The overall market has been estimated to reach about $5 billion in revenue. And our therapy, Mimetus 4555, is applicable for around half of all Duchenne patients, which is significantly more than the current approved genetic therapies that only target around 8 to 13% of patients and are just modestly effective. Mimetus has significant advantages, including that it permanently edits the DNA. It targets more patients, 50% compared to single exons, which are 13% or less. It creates one of the most functional dystrophin proteins. It edits the patient's own DNA to maintain the endogenous gene and regulatory sequences. And it retains about 3 times more of the protein compared to competing mini and microdystrophin gene therapy approaches. All of these aspects aim to make Mimetus one of the most functional therapies for Duchenne. So we have currently an exclusive license for the underlying IP from UCLA. We're currently in preclinical development, where we've shown efficacy in human stem cells in mice, the ability to redose in mice. We're located in the Magnify incubator space and funded by grants. and we're aiming to get to a value inflection point of an IND filing by the end of 2022, then progressing into human clinical trials. And there's potential for acquisition or partnership throughout this time, as well as expansion into other types of muscle diseases. My co-founders are Dr. Melissa Spencer and April Pyle, professors at UCLA. We did the initial development of this first therapy during my PhD and postdoc in their labs, and they have important expertise in muscular dystrophy and stem cells. And we also have additional SAB members who are leaders in gene therapy. We're currently seeking additional funding, either a bridge to A round to get to some value inflection points in our preclinical development, or a Series A round to get completely through it. So far, we've been funded through about 1.8 million in grants and friends and family. We're also looking to expand our team and portfolio and potentially exit through an acquisition or partnership. Help us develop these important therapies in order to save boys like my cousin Christopher, who's shown there. Thank you, and I look forward to further conversation.
Great, thank you, Dr. Young. Great, nice, concise presentation. Judges?
Yes, thank you. It was a great presentation. You mentioned, I would say, two platform aspects of this technology that I was hoping maybe you could expand on here. So one was the redosing. This is, a major problem in the field of AAVs. And then secondarily, and I think this is specific to the dystrophin, microdystrophin field, a lot of competing companies have chosen the microdystrophin, at least the way I view it is because of packing efficiency issues into the AAV. Could you speak about how you're able to pack a much larger payload?
Yeah, sure. So I'll start with your second question. So what we're actually packaging in AAV is the CRISPR therapy. So we're packaging Cas9 and 2 guide RNAs that are editing the endogenous DNA locus to remove a portion of the gene where mutations occur and allow for the native dystrophin protein to be produced. So we don't have to rely on packaging an exogenous protein where you have these major issues of fitting into AAV because dystrophin is a really large protein. So we can package basically CRISPR, edit the own DNA where we can make any type of edits and create any size dystrophin we want. And that's a really big advantage. We think it's the second generation of gene therapy, editing the patient's own DNA. And yes, exactly. Your first question regarding the idea of AAV redosing is really important. So we've been testing different approaches to dampen the immune response in order to allow for redosing of AAV CRISPR in mouse models given systemically. And we've shown proof of efficacy that this can work in these models with different combinations of immune suppression drugs. And in theory, this could be applied to lots of different types of cargo, especially because Cas9 will be more immunogenic than some other types of cargo anyways. And we think this is important for both efficacy and safety reasons. Although CRISPR is permanently editing the DNA, you still have potential to lose some of the corrected nuclei in a tissue like muscle, which is very regenerative. So being able to redose it can improve its efficacy and also help overcome some of the safety issues with high-dose AAV gene therapy that are likely caused by an immune response to AAV. If we're dampening that immune response, we think we'll overcome some of the safety issues that have arisen. And so, yeah, in theory, that could be applicable to lots of different types of AAV gene therapy. And then the idea of using CRISPR for muscle diseases by removing regions of a gene could be applied to lots of other types of muscular dystrophy that we plan to build out as a type of platform approach as well.
Thanks a lot, Courtney, for the presentation. I also went to UCLA for my PhD, so again, good to see schools representing. So I know you guys are working on figuring out, what the expression level, you need for an effective therapy. Can you talk a little bit more about, what you think that level would be and how close you are to achieving that?
Yeah, definitely. It's a really important question. So the good thing about DMD is that when you restore the dystrophin protein, it's known that it doesn't need to be restored to full 100% wild type levels. Even as little as 1% dystrophin can begin to improve function. We think to fully restore function, it might be more on the order of 15 to 30% dystrophin. And what we've been seeing so far is if we give a single systemic injection in young mice, we can see restoration of up to about 60% dystrophin in the heart and around, sometimes around 20% in some skeletal muscles. With a single injection. And we're hoping with redosing, we could even improve that level. Or if we begin to see a loss of dystrophin over time, you could then give another injection to kind of rebuild up that level of dystrophin that's needed to protect the muscle. In kind of older mice, we see less AAV tropism. So I think the limiting factor is still delivery, and it's a big problem for the field, but we can get, occasionally like 20% dystrophin in heart, which is what we see most well targeted, which is still expected to have a functional benefit. We've done some preliminary functional studies just in short-term studies in these young treated mice, and we begin to see improvements in things like muscle strength and how long they can hold onto a wire or how far they can move in an open field test just with this single injection. And we're beginning these redosing experiments over a long-term study to really look at the impact of giving more than one injection.
Thanks a lot.
I guess how does this compare to what's used right now with, I don't know if it's like protein replacement, it's probably not the right word, but for the lack of my PhD, that's probably the word I'll come up with.
Yeah, no, that's good. So the only approved therapies in Duchenne that restore dystrophin are exon skipping therapies. So these use a small oligo to reframe the dystrophin transcript on the mRNA level. And these have shown modest efficacy. They're restoring about 1 to 3% dystrophin protein, which can improve function, but it's not super dramatic improvements. And it's only applicable for very small subsets of patients because they're targeting one exon at a time, so 8 to 13%. So the big advantage of our type of approach is that we're permanently changing the DNA. It'll be much longer lasting, and we're targeting immediately half of all patients with a single therapy by encompassing kind of a larger region. That's a hotspot approach. In clinical trials, there are other approaches like the microdystrophin gene therapies. These are limited by the carrying capacity of AAV, what I was talking about earlier. So they can only package these very small versions of dystrophin. They're about 1/3 the size of normal, whereas our approach retains about 87% of the protein coding sequence, including additional functional domains, which are important for dystrophin's roles. And so we think that our approach is going to produce a more functional protein. And by editing the endogenous gene, we maintain the regulatory environment around that gene. We're not relying on an artificial overexpression that you get from gene replacement, where it's lasting as an episome, it's not permanent, and it's not in the natural locus.
And are there other people approaching this with CRISPR as well? And how are you in that stage of kind of being first to market or beating them out?
Yeah, definitely. So there's definitely been interest of CRISPR and DMD. The focus from other companies have mostly been on single exon targets. So for example, Exonics, which was acquired by Vertex, is developing what they call a single cut strategy for Duchenne. It uses one guide RNA, but the limitation is that it can only target one exon at a time. So at most, it'll be applicable to 13% of patients with each single drug, and then beyond that, it gets even less and less patients over time. And this will make different dystrophins every time because the patient's underlying mutation plus the exon that's being skipped results in these internally deleted dystrophins that vary because of that patient mutation. But our approach, because we're deleting this region, we always delete exons 45 to 55 for any type of mutation within this region. We always make this internally deleted dystrophin that lacks those exons. And this specific dystrophin has been associated with one of the mildest forms in allelic disease called Becker muscular dystrophy, where you have an expression of this in-frame dystrophin, and that has a much milder phenotype. So specifically patients that lack exons 45 to 55 have one of the mildest phenotypes. Some of them have been asymptomatic until their 60s. So we think that encompassing more patients and making this dystrophin that always has this same deletion is expected to be one of the most functional dystrophins are important advantages. And then we're incorporating these ideas of redosing, reducing the immune response, and inactivating Cas9 to deal with some of the safety aspects that other people aren't currently incorporating in their gene editing approaches.
Great. Excellent. Well, thank you so much for the presentation, Courtney, and best of luck to you and the company.
Thanks so much.
Great. Next up, we have OncoXome from Pasadena.
Hello, everybody. All right, so I will, I'm Orion Gamelin, co-founder, COO of OncoXome.
And I'm Eric Gamelin, MD, PhD, and co-founder and CEO of OncoXome. Nice to meet you.
Nice to meet you all. So I'm going actually to share my screen here for our video. There you go.
OncoXome's objective is to bring to patients with advanced cancer an innovative multi-target therapeutic approach, hacking cell communication and using gene editing to overcome cancer resistance. We have 3 platforms: gene editing therapy with CRISPR-Cas9 knocking out KRAS, our delivery system with heterologous off-the-shelf cancer exosomes, and the exosomes large-scale manufacturing.
Our first target and indications of focus are advanced KRAS-mutated lung, colorectal, and pancreatic cancers, equating to 3 million new cases per year globally. The reason behind this first selection is that these tumors not only harbor a high rate of KRAS mutations, but their 5-year survival rate remain extremely low, 3 to 14%. In that sense, we address a critical unmet medical need.
Some data we wish to share with you are proof of concept for using oncogene addiction. We transfected 2 KRAS mutated cell lines with CRISPR-Cas9 with 95% efficacy and achieved over 90% cancer cell death after single exposure. Death was restricted to cells harboring the mutated target. We are able to knock out any of the 14 known KRAS mutations. There are 2 KRAS inhibitors in development from Amgen and Mirati that only target one mutation, KRAS G12C, with some efficacy in lung, but not in other tumor types harboring this same mutation. A team at MD Anderson is starting a first-in-human study in pancreatic cancer with siRNA targeting KRAS G12D and using exosome from normal mesenchymal stem cells. With our multiplexing approach, we target all KRAS mutation and the cooperative genes involved in acquired resistance. We use cancer exosomes to specifically deliver their payload into the cancer cells. We expect much less mechanisms of resistance and adverse events.
Today, we're raising our seed round at $4 million for 24 months runway. We will be completing 4 milestones: our gene editing techniques, the exosome isolation and loading, the payload delivery, and the in vivo studies for which we will assess the efficacy and safety in mice models. By the end of the 24 months runway, although a potential exit may take place, we will have raised our Series A round in order to carry out until the end of phase 1 clinical trial. We're 3 co-founders. Dr. Eric Gamelin, MD, PhD, has spent over 30 years in oncology, gene therapy, cancer resistance. He has led a genomics lab and co-founded several startups. Myself, Orion Gamelin, has a decade of operational experience in Fortune 500 companies, and Dr. Haynes, MD, PhD, has over 30 years of toxicology, safety, and regulatory experience. And we thank you for your time.
Great, thank you very much. Would any judge like to go first?
I'm very interested. Thank you for your work. Cancer and pancreatic cancer particularly is a high unmet need. So I'm very curious the potential FDA opinion on targeting multiple siRNAs in one payload versus it sounds like MD Anderson's going after just this G1D, and that seems kind of core. Is that a fair assessment? And I guess what do you anticipate have you had a pre-IND, or what do you anticipate the FDA's opinion on having multiple kind of payloads in there or targets in there?
It's a very good question. We don't use siRNA. We use CRISPR, and the MD Anderson they have to limit with siRNA. You are limited in your targets, so that's why they target G12D. But with our approach, we multiplex different guide RNAs with one Cas9, and what we plan to do is for the submission to the FDA is to have one Cas9 with multiplexing different guide RNAs for the mutated genes, most often with pancreatic cancer. So it's the same Cas9, but what is very interesting is that you can load Cas9 with different guide RNAs with the multiplexing approach. So we don't expect any specific pushback from this approach.
Thank you.
Thank you for the presentation as well. It was very helpful. My question is on broadly dosing here. So one of the challenges in the siRNA and ASO field against oncology targets and potentially also for CRISPR, although there hasn't been as much clinical data there, has been with dosing sufficiently highly to get coverage of all of the tumor cells. How do you view that, and how do you view what potential dosing you might need to have here to get complete destruction of all the tumor cells?
It's another very good point. But in fact, it's not different from an inhibitor. We need the exact dosage. We don't expect to use one injection. We will need to do several injections, and we based on what we saw, it should be every 3 weeks an injection of the 1 million exosomes. What is interesting with CRISPR, and it's totally different with siRNA, we don't need to, I would say, overwhelm the messenger RNA. We need to cut the gene, so we need much less loading of the CRISPR than we would need with siRNA. siRNA, it's the messenger RNA. We cut the mutated gene. So with the efficacy, we expect it. We didn't compare with siRNA, not yet, but we expect much less dosage of CRISPR with this approach.
Great presentation. Can you guys, I guess sort of related to Seth's question, can you speak to the work that you've done so far on getting your therapy to effectively penetrate solid tumors? Because I imagine that's got to be a challenge to overcome.
Okay. I'm going to start with the challenge. We don't expect the same kind of a challenge as we could expect with I would say an inhibitor. We use exosomes, and the exosomes are, programmed to get to the target cells. So in fact, they avoid the immune system. They are not immunogenic. They directly go to the target cells, so they avoid the destruction in liver, in Kupffer cells, and they have a much longer half-life. Now, in terms of in vivo data, we are not yet there. We are at the in vitro step. So I cannot show you some in vivo data yet.
Well, I'm seeing some interesting comments in here. One additional suggestion that could be interesting to explore is pancreatic cancer. I've seen one company that's got a drug device combination, uses a balloon catheter, and this may be amenable for specifically pancreatic cancer, where you can kind of bathe the tumor in your therapy. So you may have less issues with systemic delivery and may be able to give a higher dosage. Just one thought to share, and I'll stop there.
Thank you.
Thank you.
Great. And we did get a question from the audience. What is the species origin of your exosomes? And a follow-up to that is whether you've characterized their tropism. And I guess the final question there, what is the largest scale you've produced them at?
So the species is human, but we don't need to be autologous. It's totally different from, for example, CAR T cells. The exosomes are not immunogenic. So we use human cancer cell lines, but we need to use the same origin of the cell line, meaning if we want to target pancreatic cancer, we need pancreatic cancer cell lines, because these exosomes will go to the same, I would say, phenotypic cell lines. In terms of large scale, we are working on in our platform, and we are using a specific approach with spheroids to be able to produce large scale, so mass production of exosomes with spheroids. We have a manufacturer, and so it's what we are working on.
Great, excellent. Just reading some of the chat. There's a suggestion for a collaboration with the first presenting company, Ericari. Chris Hughes suggested you all connect, maybe may be able to help you out. So if you have a moment, maybe you all could connect offline.
If I may, I see another chat. It's tough to get liposomes to cross the vascular barrier, and it's true. With liposome, you don't get to the brain. It's totally different from exosomes because liposomes are foreigners.
They're much bigger. Yeah.
And it's, we didn't show it. It's published. We know that exosomes cross the blood-brain barrier.
Fantastic.
Thank you.
Okay. Any final comments from any judges? I think there was quite a few there, quite good comments. Okay. Well, thank you so much, team. Very fascinating technology. Exosome space is really hot, and best of luck to you.
Thank you so much.
Great. Our next company is Surpass Biologics from Skysong over out in ASU.
Indeed, thank you. And hopefully we're audible. I appreciate your time and consideration today.
You're good.
Thanks. Awesome. To challenge the status quo and drive innovation in medicine, we have synergized millions of years here at Surpass with the development of modern pharmaceutical engineering to create a novel and potent drug. This is both a hallmark of our team and our primary asset, which are built upon considerable backbone of our first generation protein therapeutic. This biologic actually has already shown both safety and efficacy in the clinic. Today, we're going to be presenting the next gen potent anti-inflammatory based on harnessing the power of that which has been done before. This is our motivation, and we are the team driving innovation in medicine. My name is Mila Chikara, CEO for Surpass Biologics, and I have with me on the phone today, Alex Lucas from ASU. Inflammation is the immune system's normal reaction to both protect the body from injury and infection. However, chronic, overstimulating, and self-injuring inflammation is a main driver of disease. Vascular inflammation in particular is a pathological process that occurs in many overreactive and autoimmune disorders. And this is exactly what we are targeting. Blood vessels become inflamed. They're either leaky or clogged, and as a result, often damage the organs themselves to which they lead. It is for this reason that out of control vascular inflammation is debilitating. And our current standard of care exposes the lack of both available and appropriate therapy. Our solution is an anti-inflammatory, targets these areas of overreactive, autoreactive, and damaging inflammation. In contrast to standard of care, AKA throwing steroids at everything to work against the immune system, our protein therapeutics work with the immune system in a targeted and potent mode of action. They block damaging and out of control immune responses, all without suppressing protective immune function. This biologic is a SERPIN, shorthand for serine protease inhibitor. Our unique biologic SERPIN, SPS-101, is an engineered version of our first generation therapeutic SERPIN, SERP-1. SPS-101 is a highly potent immune modulator, bringing both control and balance to an out of control immune system. Over 40 million patients experience debilitating inflammation. To facilitate our go-to-market strategy, our plan is to address unmet medical needs in the rare diseases space with precision medicine, thereby reducing hurdles and increasing our velocity to market launch. Our protein biologics competitive advantages will enhance healthcare providers' options of a highly potent and targeted anti-inflammatory. And with evidence of restoring proper immune function and safety data, this allows us to get to IND clearance as our next inflection point in 24 months. In summary, our goal is to raise funds. In order to innovate medicine, we'll rival the standard of care in an exceedingly large market, are backed by clinical evidence, and are well positioned to be first in a new class of anti-inflammatory therapeutics. In summary, Surpass Biologics takes out of control inflammation and brings it back under control to a healing state. Thank you, and looking forward to questions.
Great. Thank you, Mila. Would any judge like to go first?
Oh, thanks for the presentation. Can you guys speak a little bit more to the precise mechanism by which these engineered SERPINs are modulating the immune system?
Yes, so the exact mechanism is as a serine protease inhibitor with a UPAR. Alex, would you like to elaborate there a little bit?
Sure. These proteins have evolved extremely well, and that has made them very potent. They target both sides of the protease cascades in clotting and also clot dissolving, thrombosis and thrombolysis. And in fact, they are not enzymes, they are proteins. So they only go where there's activation and excess activation of one of these pathways. Both of those pathways are highly active in immune and inflammatory responses. And we've tracked it all the way down to the UPA receptor, which is now known to be inflammatory. So we found that we can use these proteins anywhere from picogram to microgram amounts from our early studies. They're fascinating because they're so highly effective. And to reiterate what Mila has said, we did go through clinical trial with our first generation, no side effects. It was in people with coronary stents for inflammatory atherosclerosis, no neutralizing antibodies, which is amazing. So they're built to be effective at low dose and with minimal adverse effects. So now that we have a next generation, it's brought us up into a new competitive advantage where we can actually go forward and have IP protecting us. And our first studies have actually shown benefit over and above the original in animal models. We haven't been to people yet. We got to get there. So does that explain it well enough for you? Yeah.
Thank you both for the presentation as well. Maybe you could speak to why the first generation molecule was discontinued and how the new generation molecule is solving whatever those challenges were there. I'd say that that's probably a good place to start. And then secondarily, and this is a separate question, why are serpins the right modality to go after this receptor? It's fairly well trodden the path of making an antibody that inhibits or blocks receptor activity. So maybe you could speak to that as well.
Sure, I'll tackle the first part about it. Why re-engineer the first generation? In general, protein therapeutics can always be improved. What we did was actually set out that first generation and extend its half-life. This was actually going to be able to allow us to expand utility for indications. So really our primary objective was to optimize the PK/PD to expand clinical use. It gives us ultimately flexible treatment in regimens and indications, but it also came with some very strong positives. One of the main positives was that the strong preclinical efficacy with delayed timelines was remarkable and incredible. And I see my screen isn't shared anymore. I can put up a slide generally like that if you would like to see that as well. I don't know if my share screen function still works, but I can certainly try that. And interestingly here, when you look at this, plus brand new composition, 20-year matter, composition of matter IP, we plan to exploit this all moving forward. So that was the scientific basis of this. And the reason for viral-derived proteomics versus monoclonal antibodies is really due to the power of efficacy and ability to immune modulate without the side effects that we've seen traditionally in monoclonal antibodies.
These virus-derived immune modulators have been evolved for us. The R&D was done by the viruses and done very well, and they're not regulated by mammalian control systems, which has been perfect. It's actually left us with a highly potent agent, and yet in all of the models so far, no side effects. Why did we not progress? We were old as we got started. We got started many years ago in a small lab in Canada and a smaller company in Ontario. And by the time we got moved forward, it was already getting to be older, whereas now we can start again with all of the efficacy and safety proven with an even more potent molecule. So we're quite pleased with it. And it is a new class. Serpins are used, mammalian serpins, but again, they have all of the attendant problems of a ready-made mammalian protein and that they will be regulated by our own mammalian systems. So we basically, as one of our former leaders in this work, we've harnessed the evolution of millions of years of virus development, and they're really potent. So that's about all I can tell you so far. Did that answer the question for you?
Yeah, thank you. Although maybe I'll just re-ask the first part of the question, which is why was the original SERP1 discontinued? And I grant that better PK and better dosing can be more convenient for patients, but if there's an efficacious treatment out there for grievous disorders, a once daily therapy is not out of the realm of question. There's plenty of examples out there. Insulin's, I think, most famous one where you have daily, sometimes multi times per day treatment. So I'm curious as to why the first generation was discontinued rather than continue clinical development.
Oh, we didn't discontinue. We just needed to raise funding and we weren't able to get enough IP with an old protein. We wouldn't have been protected with patents. Now we have protection and we have actually improved efficacy. We still use our freezers full of the GMP quality protein and it is highly effective.
Correct.
Yeah.
Yeah, and that's one of the things we have our master cell bank that all of that first generation is based on. So that's going to be a very fast improvement because our re-engineered version is used off of our previous GMP material. So yeah, sorry, it's not discontinued. It was simply improved upon.
Yeah. We have to master cell bank as well.
Correct.
Yeah.
Great.
This is helpful. I guess speaking from the angel lens, when you look at something like this, and this also applies to some of the earlier presentations, you're looking for bigger capital inflows. Can you speak to, we didn't speak to this with some of the earlier presentations, but what are your capital needs and, where are you looking for that to get you?
So the capital needs presently are 8 million is what we're asking for in a seed round. And that's going to take us through our next inflection point, which is IND clearance, and we're expecting that to take 24 months. Again, because of the re-engineering with the pegylation of the original first generation, that's going to require a revamp of the IND enabling studies. So that
I guess
effectively
just to poke a little bit on that, okay, do you have a breakdown of like what's behind the 8 million? And if you already had kind of GMP quality manufacturing, did you guys raise 8 million previously for the, seems like pegylation added to the original wouldn't be that much more expensive, but I'd love to kind of see the breakdown. Maybe I'm missing something.
So the original publication, which we can share, was a little bit more than a decade old now with respect to that first generation clinical trial and molecule. Currently now we're going to have to effectively re-administer our pharmacology studies, which include mouse PK work, the mechanism of action in vitro, to prove that it's working the same way even with pegylation. This will also include bioanalytical detection in both mouse, rat, and we believe that monkey is going to have at least to be required there. Further, the bioanalytical assay development, PK studies, and safety pharmacology are all going to have to be redone as we see it. So that's ADAs, NABs, and bioanalytical in at least 3 species. And so seeing as the analytical BA and PK studies are going to have to be redone, so will the safety pharmacology. So we're likely currently planning respiratory, CNS, cardiovascular in rodents and canines, and then we have 14-day studies coming up for repeated dose toxicology as well. Yes, we do own the master cell bank, which is a very strong perspective of building and efficiency, but we do have to bring that back into a GMP setting for production. So we're basically going to be starting again with the formulation development, making sure that what we've previously prescribed is going to work. We have the master cell bank. We basically need to tech transfer that, optimize their new process, and then create the production process from there to ensure stability. All along those lines, we're going to be ramping up and prepping for the first in human. So clinical and regulatory strategy and implementation is part of that 8 million.
Thank you. And I guess going back to the pathophysiology of this, you have a lupus model here. Does it reverse kind of some of the progression, or is it more kind of preventative as the patient's getting worse?
That's one of my favorite topics. Mile can present it too, but we had always treated at the onset of disease in all of the models and our collaborators. But we found that with the longer half-life and the new modified protein, we can wait 7 days and then treat in the mouse lung hemorrhage model and still see marked efficacy, which actually I wasn't expecting, so that's a bonus. So not only have we increased the half-life quite a bit, but we also now see that we can treat after 7 days, which would be closer to what you'd see in clinic. When we see patients come in clinic, they are often already in the throes of, say, lung hemorrhage or acute vasculitis. We would be able to treat and have expectation of benefit, which is huge. And
Yeah, this is often,
go ahead.
sorry, Alex,
No.
just to reiterate there, often inflammation is so general. When you show up to a healthcare provider, you are on a diagnostic odyssey, and that odyssey can take days and even a week. So while many patients are experiencing very debilitating inflammation, this experiment here also shows that you're mimicking a true clinical setting where treatment therapy is delayed, and you get quite effective evidence of restorative function, both of the vasculature, homeostasis, as well as organ function.
Great. All right, great. Well, Mila and Alex, great presentation and responses to the questions, and best of luck to your company.
Thank you.
Thank you very much. Appreciate the time.
Yep, thank you.
Thank you all.
Okay, moving on to our last presenting company for this first session, Tortuga Biosciences from UCSD, coming all the way down the coast.
Hi, thank you. I'm Ted Daly. I'm going to talk about Tortuga's precision microbiome therapies. Microbiome is an active area of research and development. Companies and investigators are testing whether live bacteria can be used to treat a variety of diseases. In some cases, these bacteria have been genetically modified to express beneficial functions. However, most microbiome therapies have not shown much, if any, actual effect. And a key problem is that foreign bacteria just can't compete with the host's native flora. So an introduced bacteria doesn't survive long enough to really have any lasting effect. Consequently, most microbiome therapies in development require frequent redosing, often multiple times per day. Tortuga's innovation is engineered native bacteria. So it's analogous to CAR T, except that instead of extracting T cells, we're extracting bacteria from the host and engineering in a beneficial function, culturing the bacteria, and administering it back to the host. And because it's already adapted to the host's microbiome environment, that engineered native bacteria can survive and can colonize and have lasting effect. We've proven that we can attain long-term colonization in mice. Here we show that mice that received engineered strains of native bacteria, the bacteria was detected for 300 days after a single administration. For comparison, non-native bacteria introduced into a mouse only are detectable for about 48 hours. But more importantly, we've shown that we can achieve long-term physiological change. So in this example, mice that received bacteria that had been engineered to express an enzyme called BSH showed improved glucose control 15 weeks after a single treatment. Our strategy is to develop this technology for treatments for metabolic diseases. Our lead program we plan to target is PKU, a genetic defect of the enzyme that breaks down dietary phenylalanine. PKU results in developmental delays and intellectual disabilities. The existing treatments for PKU are chronic daily therapies with challenging side effect profiles. The projected market for PKU therapeutics is expected to grow to nearly $2 billion by 2025. And we believe that a long-acting alternative to the chronic daily therapies can capture significant market share in this patient population. There are a number of other orphan metabolic diseases that we plan to target in similar patient populations that collectively represent a multi-billion-dollar commercial potential. And while we focus company-sponsored development on rare diseases, we plan to partner with Big Pharma to develop the technology for treatments for more common diseases. We're raising a seed financing to conduct some key proof of concept and translational studies. These include demonstrating efficacy in a PKU mouse model and establishing the engineered native bacteria platform outside of mice in an environment or in a system more representative of humans. Initially, we plan to do this in pigs. And then some additional translation studies that will prepare us to enter clinical development. This will be over about an 18 to 24 month period. With that, I'll leave the background on the founders and our contact information, and I welcome questions. And my colleague Amir, and he's founder and inventor, he's trying to join, and he said he had some connectivity issues, and so I'm hoping he'll be able to join for questions, although I'm not sure he's going to be here.
Great. Thanks so much, Ted. Good to see you, and congrats on this venture. Would any judge like to go first?
Yeah, I'd be happy to go first. Great presentation, Ted. So I was curious if you could tell us a little bit more about the level of expression of your target therapeutic protein in the studies that you've done before. It's good that you have persistence of the bacteria, but I'm assuming there's some component that it's emitting that really is having the therapeutic effect for PKU or whatever indication you guys are targeting.
Amir, I see you on. Are you able to, can you unmute and you want to address this question? Amir?
He's unmuted, but he may have sound issues.
Okay. Yeah, unfortunately, I think I probably have Amir address that one.
Okay.
Oh, no, I thought he was in.
Wouldn't be a Zoom call these days without some technical challenge.
Well, he's okay. He's now, I see him on the panel here. Amir, are you able to
answer
that question?
Amir, your microphone may not be working.
Okay. No.
Yeah, no worries. We can come back to it if the sound comes back.
We almost made it through the first session. We have another question, perhaps on the market. It might be interesting to Ted. I think that's probably more what you've thought about here.
Yeah, so maybe I'll ask a sort of more market-centric question. Given that this would be autologous, it sounds like, have you thought about what the COGS would be? And then it's probably not a one and done therapy, so there's probably some redosing at some point necessary. So how do you view that? And then how large of a dose do you need to dose here? So sort of baking all those together, essentially what I'm trying to understand is relative to how much you're gonna have to spend on this therapy, how much will the market be willing to bear in this situation?
Yeah, I mean, it's an important question. You're correct in that we don't know if this would be one and done, truly curative, or more, or possibly it would be something that needs to be re-administered periodically, perhaps like on an annual basis, and that's going to drive the commercial model. We're also exploring this in addition to autologous one-to-one, more of an allogenic one-to-many, which would drive the cost of goods. If it's in the autologous setting, it would be the isolation, engineering, and cultivation would be done in a specialty lab, and it would be administered either as a lyophilized bacteria or a slurry back to the patient. In a one-to-many, more allogenic setting, it would be the isolation and engineering could be done in a specialty lab, but there are manufacturers, third-party manufacturers, that are doing more large-scale production of live bacterial therapies, and it would probably be in a lyophilized bacteria in a capsule, for example. So we don't have specific costs from a revenue standpoint. If you just look at marker or benchmarks for gene therapies and cell therapies, there are some benchmarks you can look to for how this might be priced if it's truly a curative one and done model, or if it's something that is re-administered periodically. Again, there are precedents for these rare diseases or chronic therapies that serve as commercial benchmarks for how we would, how we see pricing these treatments.
I hear hospital background sound. So I'm
Yeah, can you guys hear me?
I
I think I got the audio to work. I think there was something conflicting with it.
Yeah, we can hear you.
Yeah.
Hey, somebody want
I
to ask that question again? Or Amir, did you
Yeah.
get the first question?
Yeah, absolutely. So generally, I was just curious about, the level of expression of what I assume it's a therapeutic protein that you're expressing in these bacteria. How persistent is that expression, not just the persistence of the bacteria itself?
That's a really good question. We're still trying to characterize that and figure that out. Sorry, I'm still at a hospital, and this was arranged during one of my clinic days, so I really
No
apologize. I
problem.
really wish I could be
That's
giving
okay.
this my 100%. But I think what we're trying to do is what we've been doing is collaborating with a colleague who has been able to help us characterize how much we're changing the functionality with this bacteria. And what is absolutely remarkable is that compared to some of the engineered probiotics that are out there, those people are, some of the technology that's been described have been getting 10 to the 12, so 6 orders of magnitude higher than what we were getting with our engineered E. coli. And but we're seeing a 3 to 4-fold change in bile salt hydrolase expression in the lumen compared to what they're seeing is less than a 50% change in GFP expression. They haven't really shown whether they can change function in vivo yet. So it's kind of hard to do an apples to apples comparison. But even in germ-free mice, so in reduced community mice, what we're showing is that these bacteria, since they're fully adapted to live in that luminal environment, it doesn't hurt them that much, or it's not that more difficult for them to express a transgene that may not be to their benefit as it would be for a bacteria that now has to figure out a whole complete new strategy to survive in this foreign environment and to express a transgene that is not to its benefit at the same time. So I'm not sure if that's getting to the answer to your question.
No, that's
the other
very
thing that's a
helpful.
little bit of a caveat here is that these, and which other people aren't really reporting about their bacteria, is that we're finding that bacteria levels and expressions change with time, which is something that is, and when I mean time is within a 24-hour period, just because there's normal circadian changes with the microbiome. What we're finding is that this bacteria is expressing this 24 hours. It's just that it's having most of its beneficial effects during certain windows of time. And other people aren't really reporting these kind of things with their bacteria. So I think what we're going to find is that engraftment is going to become more and more and more important. And that's why we're seeing a lot of failures with early engineered probiotics, is because they're not really managing the time in which the probiotic is going to be most effective for its therapy in terms of what's going on with the host. So you probably need a bacteria that is delivering the function at the specific time that the host needs it. So, so that's going to be another element that we're going to see in the future in microbiome research.
Thank you.
For this, this goes to Chris's question a little bit in the audience here. He asked if the engineered I have 2 questions. So one is a safety question, which is, what safety concerns might there be? And then to Chris in the audience from UCI asked, how do the engineered bacteria have or need a kill switch, which kind of goes to the safety question. What safety concerns do you see for this potential, gain of function engineering? The other question that I have is, with PKU as your lead indication, I did a quick search and based on my quick search is it maybe a monogenic disease that may be targeted with CRISPR, which may be a highly competitive field. So I just wanted to also kind of poke on, why PKU first and maybe not something else that may be more, specific to the gut.
Sure. So,
So quick question, quick answers about safety. I can answer quick questions. The quick question about safety. We have now given engineered bacteria, both with bile salt hydrolase and with other genes and alone, for now I think we're over 1,000 mouse years of experience with these bacteria. We've never seen a detrimental effect. We have 87 weeks of bile salt hydrolase expression with no detrimental effect in mice. And these are bacteria that are already there. So we're basically, these bacteria are essentially part of the community. So the fact that somehow, I think, with FDA approval, each transgene is going to require its own analysis as to whether the function that we're expressing will be detrimental, but the bacteria is already there as part of the community. So the bacteria itself, the vector doing something detrimental, is highly unlikely. And then in regards to the second part of your question was related to, could you repeat that second part?
I think it was about CRISPR.
CRISPR and competition.
Yeah, with PKU specifically.
Yeah, so and kill switch. And so I think the reason why we went after PKU is that it's a function that you'd never want to shut off. It's basically a function that the patient is going to need all the time. So so that's part of the reason that we thought it was a low-hanging fruit. The CRISPR tells you how you can get the gene into the bacteria. What our IP is on is the strategy of using autologous or allogenic use of a native bacteria. So you can use CRISPR, and you may need to use CRISPR to get your transgene of interest into undomesticated native strains. And in fact, I think but the novelty here is the autologous use of something that already belongs to the patient or belongs to PKU patients in general. And that's what's novel here and what the IP is built around.
Great. Very interesting presentation, Ted and Namir. Congratulations on all the success and best of luck. I think that brings us to near the end of our first session. So Andy and Steve asked if I had any announcements myself, and perhaps one or two brief ones. One is that each November, December timeframe, Q4 really of each year, my firm Wilson Sonsini hosts a therapeutics life science investor forum. And traditionally we've focused on therapeutics, but we've also included genomics and synthetic biology. What everything that has been presented today would qualify. So keep that in mind and feel free to contact me as we get closer to September timeframe. We tend to have about 50 to 60 life science focused VCs, seed, Series A, later potentially of interest. And we just play matchmaker and connect the funds with the companies. Ends up being about 300 companies submit, and a good portion of them do get meetings. So keep that in mind. And then the other plug, if I can help you in any way, let me know. I'm a life science patent attorney, biochemist background. Lots of very interesting technologies I've heard today, and do a lot more than just patent. Really do a lot of connecting with investors and helping strategize on IP licensing and enforcement. So feel free to reach out. So with that,